Here I am again, Gilberto Thomas from Brazil. And this lecture is a kind of continuation of the first one that will treat with the properties and behavior of the cells. In particular, we are going to analyze how to make a cell grow and a cell divide. Okay, how to use some specific steppables from CompuCell that does those processes for you. Remember that the workshop is being live streamed, recorded, and distributed on YouTube. Okay, let's start by showing the topics of this talk. I will start with something about cell proliferation division, about cell cycle, I mean the biology of the thing. Then I'm going to move to the implementation of growth division in CompuCell. And I will follow some 3 examples where I'm going to show you how to in fact, implement cell division in CompuCell. Okay. Growth and division in three examples, including the last one when we're going to treat with contact inhibition growth, when the cells doesn't have more space to grow and divide. Okay, let's go. Starting with this basic is biology about cell proliferation division. Okay, so when the cell starts to decides to uh, divide, it is a consequence of a bunch of process inside the cell, and this process are usually uh, dividing in 4 categories like proliferation, survival, differentiation, or death. Like Professor James talked this morning with a bunch of questions about how the cells do that. In fact, we know a little about this process, and he showed those pictures as well, this picture as well, showing the intricate chain of events, of chemical, of interactions between the cell and the extra cell, in interactions inside the cells, inside the nucleus. It's a very complicated uh, way. So, for example, for proliferation, we have interaction of growth factors, which usually comes from the external environment of the cells. We have cell-cell contact, which can inhibit the proliferation because it occupies the space that the cells would have to proliferate. And we have a variety of other factors that can promote these 4 outcomes. And each one of these outcomes depends on this cell state of the cell. Okay, let's understand what we can say by cell state. Okay, when the cell decides then to proliferate, it enters the cell cycle that it repeats itself from time to time. For here, I can show schematics of this cell cycle. The cell cycle has 2 main phases, let's say that way, which is the interphase, which is compounded by 3 secondary stages, G1, G1, G0, 4 actually, né? G1, G2, G0, G1, G2, and S. And the final one is mitotic phase where the cells really divides after pass through all these stages here where is a preparation for the mitosis occurs. So it's a long and complicated process, and a good model should be able to reproduce that. Uh, Also, the cell cycle includes some checkpoints. At, at the end of each one of the intermediate state, G1, S, G2, and M, there are checkpoints when the cell tests if it is ready to go to the next one. If the cell fails in that test, it dies. I mean, it produces a apoptosis process and the cell dies. Mhm. Well, so to model everything is a huge task. I don't think it's possible so far. But we can implement some events on some mechanism to start. The simplest one, which define the division, is the growth and the division itself. And to go to this stage, uh, we can make us try make a to answer some questions, like Professor Grace said this morning, to what can affect the growth states of the cells. For example, do they have enough space to grow? Do they have enough nutrients to take up and increase its size, or to have energy to divide? How fast can the conversion of nutrient into cell mass a cell can make it? So if there are growth factors in the middle to promote the growth. So it's a bunch of things that are involved in those processes, which is this is why the task is really hard. But we can begin with simple simple rules will that we implement to the growth of the cell. These simple rules is the following. Let's say that the easiest approach to, for the cell growth is to increase its target volume through a constant amount, say it's G here, at each time step or each amount of time, interval of time steps. So the cell increases targets, so the actual volume will increase accordingly with target volume. Remember what we have seen in the previous lecture. Okay? So the question is, how do you implement this rule in the simulation? Okay? The volume plugin, when you load the volume plugin, it comes like that, just a line in XML that doesn't have any other instruction. The instruction about the volume, the target volume, and the lambda volume of the cells are given in the steppable file, in the start function of some class in the steppable file. And the target and the lambda are defined by cell, cell ID, as they say here. Okay. Remember, the cell has two main identifications: the ID and the type. We are talking here in cell ID. ID is a number. Type is the cell is mesenchymal, is epithelial, is whatever, nervous cells. Okay? The type is a number. It's a unique number that identifies one cell. So we attributed in the Steppable, as we saw this early today. And in the step function, we implemented this kind of growth which we have here. We run over the cells here in the cells which is already created in the start function. And for each Monte Carlo step or a certain amount of Monte Carlo step, we increase the target volume for certain value. Here, for example, 1.101 pixel per addition. So it takes 100 Monte Carlo steps to a cell to the target volume increase by 1 pixel. Okay? It seems small, but it's not. So once you have this implemented, let's see how to implement this in the code now. Again, I will give you the roadmap, and you go code yourself, as we did in the previous action. Okay, so start this new project in TwoEdit with here's the roadmap. I will go step by step to show how to proceed. Okay. Again, the code is in the zip file in the student material demo codes. Let's go here. Again, just being repetitive, but at the beginning doesn't do any harm. So you open the TwoEdit, go to the simulation name, give it a name, choose the destination of the simulation, choose the Python XML file, go to properties, 100 to 100, and now let's start with a blob instead of a rectangular slab. Okay? Advance, go to the cell specification, add a single cell, create a single cell, name it cell, for example, because we need only one to start, one kind to start, and add it, and go to ignore the, oh, I see this is later, sorry. Go to the cell properties and include contact, volume flex, surface flex, center of mass, and now the novelty is the growth steppable here in the simulation. The next step is ignore the diffusion fields windows. In the contact and internal plugins, you set neighbor order to 4 and finish the wizard. It's very simple. Again, you give it a name, create a blob, introduce these plugins, load the plugins, contact, volume flex, surface flex, center of mass, and growth, set neighbor order to 4, and open the CC3D project in the TwoEdit. Okay, and go to the XML file and modify it. In the last part of the file, you have the BlobInitializer now, or instead the uniform initializer you had before, and put the radius of the blob, the working region 5, and the width of the cell also 5. So you are going to create, again, only one cell because the cell is occupying all the region. Okay. Uh, no, I don't. Well, let's see. Now we have to change the steppable. Why you have to change the steppable? Well, we have a new steppable now. To begin, just a review. Let's analyze the steppable structure. This is some steppable, doesn't matter which one, it's just to analyze the structure. We have these 5 functions here: init, start, step, finish, one stop. Each one has its particular function. This is for initialize some parameter. This is to define some initial functions. This is a step it runs every Monte Carlo step, which is defined in the main Python. And finish it if you have to include something to end the simulation, and what does the code, the simulation must do when it stopped. So these five files are here, 5 functions are here. It's not obligatory to have something in the on stop and finish, only if you need something more specific. Okay? So to modify it, we have to include now these changes. In the start function, in the first steppable constraint initialize, we include this. Well, I think 25 is already defined there, but you put the lambda volume equals to 3. Second, we have to implement now how the target volume grows. Okay, this is made in the step function of this growth steppable. In the second step, we are saying that the growth is 25 walks or pixels per 100 Monte Carlo step. So each Monte Carlo step, it will add to the target volume this amount here. And the cell will grow according with this rule here. Okay? Everybody's following? Yes, it's fine. Okay. Advancing. Just a second. Okay. Now we have a new thing. I think James has showed yesterday how to plot graphs. And Maciek also has a little addition of a plot graph window this at the beginning of the afternoon. But today I will going to tell you how to add a plot window. Okay, so look, when you load this your growth steppable, it doesn't come with the start function. It's by design. It should have, but it doesn't come with. So you have to create it. So typing in it, exactly like that, aligned with the other def above. Remember, Python has strict rules about alignments of similar structures. Okay, so def start self to comment, and go to, after you do that, you put your mouse in the next line, and go to drop-down menu of CC3D Python. it's like that. Go to the scientific plots line, click on it. It will open a second drop-down menu, and you click on this choice here, Setup. See, Setup puts only it's designed to be included in the start function of the class. So clicking there, and this window in this line is in the yellow box will appear not like this in your screen, in your simulation. And then you configure your graph. Okay, so you change the title because it comes with a default configuration. It's called average volume and surface, something like that. But you can change it. Put in the title cell volume, Monte Carlo step you keep that way, y-axis which change to volume because you want to track the volume of the cell, and change the grid if you want for true. Grid is that small lines that show up in the graph to clarify the values of the quantities of the axis. Okay, like these lines here you see in the image here, and the extra line here, self plot win add plot, is the line that creates in the window plot a placeholder for the quantity you want to plot. You have to give a name for this placeholder. In this case, this plot I will call vol or volume. You can change the color, you can change from line to dots if you want, you can change the color, and you can change the size to 5 to line be thicker, to be more visible. Okay, it is better than that way. Okay, now you have to, this is in the start function. Now you have to make the volume, the cell volume to be plotted in that graph. So you go to the step function here and make a line, include this line here, which is also from the scientific plots drop-down menu from CC3D Python table. But now you see is the second line, add data points. See, into parentheses, is step function utility goes to the step. So if you click here, this line will appear. Actually, I think it will appear two lines because at the beginning you have two lines here. The second line in the start function, you can delete it. Keep only one for now. The same thing as here. See, when I add the plot point here, I must refer to the volume, vol, which I define in the start function. So inside the placeholder here, I write vol. And I plot x, MCS, the time, and the quantity I want to see, cell volume. So this loop will show for if it has more than one cell, the volumes of each cell. Since we have only one, it's going to show only one cell, one curve. Okay? Now save the file and run the simulation. Let's see what happens. It must be very fast. This is what you should see. In the plot window, you have a straight line. Means that the growth is linear, and the cell is growing in a rounded way. Now let's see the effect of the constraints of the cell in the cell surface, actually perimeter in 2D. To do that, remember when we build the simulation, we included the surface plugin, but we didn't implement it here in the start function. We are going to do that now by hand. So you have to type here cell target surface equals to 20 and cell lambda surface equals to 3. Then we are initializing the values for surface, for the target, and for lambda surface. Okay? And we are going to see also, we want to see also both now, volume and surface. So in the start function, we add a very similar line 29 here in the screen equals to line 28, but replacing vol for surface, change the color from red to blue, okay? And also, in the step function, we add a data point for the cell surface. This way. You can copy and paste the line 35 and change vol to surf and volume to surface here, and that's okay. You can do the same for 28, 29, change vol to surf and red to blue. And run the simulation. Save it and run the simulation. So now you should have this thing. See, the volume still grows. Actually, the target volume still grows, not the volume, the target volume here. And the cell, the volume grows, but the surface doesn't allow that the shape change from the square. Because, see, the surface is fixed in 20 here. The target volume is changing, is increasing here. But the surface is not, it's fixed. So the cell cannot increase easily. The surface cannot make the curvature to be round. It have to be straight. Yes, we are ignoring the unities. We're just saying measuring the amount of pixels, let's say that way. Okay? Those are pixels now. We are not using meters or square meters or whatever, nor even time here, not second. Those are simulation units. We have more freedom to do that. So this is why the cell keeps square. So how do you correct that? We have to adapt the target surface to the actual target volume. I mean, we have to make the target surface grow accordingly with the target volume. So remember, now we are dealing with not a square initial state, but with a circular initial state. So I will approximate the surface like the perimeter of the circle, 2 pi times the square root of the volume, which is the area. Okay? And I will introduce this line here in the line 36. Cell target surface is 2 pi, and for that I defined pi before, because Python doesn't know what pi is. I have to define it here. So cell target volume is 2 pi times square root of cell target volume that I just have updated. Okay? So doing that, you should have this. Save the simulation, hit the play again, and this is what you should have. The cell now grows and rounds. So adjusting both plugins, both constraints, the target volume, the target surface, accordingly one with the other, the growth of the cell is smooth, more biological, similar. Go on. Now we have to trigger the division. So far, we just increase the cell size. We are increasing the cell size. I mean, like, remember the rules? We are allowing the cell to grow to reach some amount of mass. And when we reach a specific value, it divides. This is what we are going to do now. We are going to divide the cell. So the question is, what triggers the cell division like this? See, it's a complicated stuff. The cell evolves, it increases a little bit. You have to destroy the nucleus. Where my mouse? Oh. Well, contract the genes, separate them, divide, and recreate the nucleus. It's a nice process, actually. To simulate that it's feasible, but it's not what we want right now. We are not modeling structures like nuclei, for example. Now our cell is empty, just intracellular matrix, if you want. Okay. The simplest way to divide the cell is to say that, okay, when it reaches the double of its initial size, then we divide it. Okay? Like that, simple like that. Okay. But let's think a little bit about cell division in CompuCell, the terminology, how it works. Okay, so it's obvious that one cell is split in two. So we needed to create a new cell. I mean, the create a new cell the cell is created by the division. I mean, when you divide it, you have two cells in the simulation, but you don't have yet the object new cell. You have to create it, and create with some properties and some attributes. Okay? For simplicity, to identify who is who in the simulation, in, although in biology we cannot say that, we are going to say that the parent cell is the one which is going to divide, and the daughter cells are the product of this division. Okay? So, the decisions you have to make for the division. For example, which parts of the parent cell goes to the daughter cell? The daughter cell is really equal to the first one, for example, or it's only a part of it that goes to another cell. What is the plane of division? Like you see in this picture here, the division could be axial, could be in the middle of the cell, could be in a random orientation like that. We have to decide that. Okay, how the attributes of the parent cell are inherited by the daughters. I mean, is the daughter cells in the same cell state as the parent one, for example? We have to decide that. Everything has to be decided. It's like James said this morning. You have to think before you start to to code. After you have this pseudocode with this, okay, I want my code to do that. The cell is like that. I will have division, blah, 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 blah. Then start to code. This is a bunch of the decisions you have to make before. Otherwise, you get stuck, and as he said today, people give up to code. Okay? So let's continue. So there are a bunch of possibilities to decide how are the the divisions, the attributes for the daughter cells. Okay, well, let's continue. CompuCell has a special steppable called MitosisSteppable. This MitosisSteppable, it comes with the basic operations to do what I just said, to perform the cell division, um, triggered by some mechanism, and the basic is to pass, to inherit properties from the parent cells to the daughter cells, and to define what kind of the daughter cells are. It's a very nice step up, very useful. And the only thing you have to do by hand is to adjust targets, volume, and surface inside the steppable by hand, okay, by yourself, because you have to decide how the things must behave. So it must be by hand. As I said before, no, the question, what triggers the cell division, is a particular event. And if for us here, what is going to trigger our division is when the cells reach a double of this initial size. So implementing, if we have a bunch of cells in our simulation, we run all over the cells, interact all over the cells, looking at its actual volume, and see if this volume is ready to be divided or not. Let's see how this is done here. This is the structure of the mitosis steppable. It has this name. And in the first block here, you have a list. It comes with an empty list that is called cells to divide. Then this is a condition for to trigger the division. If cell volume reaches 50, remember our initial cell has 25 in our usual simulation, default simulation. So if the cell volume is bigger than 50, so I put these particular cells in the list to divide. Cells to divide append cell. So the cell which has volume bigger than 50 goes to the list. So interact over all the cells, and after that, I look at the list for cell in cells to divide. I mean, I'm looking inside this list here, okay? Because every cell which is here must be divided. So I have chosen here an orientation division. We could set other values choice here. You can choose a specific set of axes, major axes, minor axes, but to do this particular choices here, major axes and minor axes, you have to use well, actually, we are loading the center of mass plugin, which has been loaded already. Okay, so after this line here divide all the cells which are inside the list. After that, we have to update the attributes of the parents. The first one, since the parent was divided in two, the target volume of his must be brought back to initial value 25 or something below half of 50. Remember, we were growing the target volume each amount of Monte Carlo step. So after division, we come back to original value, divided by 2. And this command here, self-cloned parent to child. This command clone to the child cell, to the daughter cell, every attribute from the parent. Okay? Except type, which we are going to change here. See? After division, we should also define the type of the child cell. Look, in this line here, I'm saying that if the cell parent type is 1, I mean, the cell that was divided is 1, then the child will be a different type. If the parent cell is 2, which means this else command here, then the child cell is 1. It's always swapping types here. If you 1 goes to 2, if you 2 goes to 1. Okay? Understood? Okay. So let's have the cell divided and reach the W1. Okay, so we are going to choose this line here to put the condition to doubling. Now we are going to write the code. We are going to use these values here. Okay, and just to repeating the main part of the mitosis steppable, here's the cells to dividing list. Here it iterates over all cells, and if the cells goes to over 50 in volume, they divide. That's this. To perform the division, we have this, just repeating everything in clarify modes, duplicating. Actually, I should go faster just to code. Choose one, self-parent, and parent child. Okay, now I need to, this is that why I run, because I would like to get real, because this is an important thing in CompuCell. There is a Python issue because if you define quantities in one steppable, you cannot have it used in another steppable. Okay, it's not Fortran or C that you can define global variables that can be used anywhere in your code. No, here if you have a class, everything you define inside a class cannot, in principle, cannot be used in another class. So you have to use some special function, Python function that are dictionaries, which have this form is between keys here, and dictionary is a collection of key between commas here and values here, separated by 2 points. The keys are immutables. The values can be changed. Okay? And the access to a particular key value pair inside the dictionary is like that. Print, for example, my dict name. The output would be the value of the key name. So it's Alice. If you want to modify a dictionary, you attribute a new value to him. For example, here, age is 30, but you want to change it to 31. Just say my dict age equals to 31. This will change it for 30 to 31. We can add new keys to the dictionary. For example, here, my dict, a new key, because email didn't have in the former definition. You add a new key email with value at x.com, and this will be added to the list, to the dictionary. If you want to delete some items of the dictionary, you say del my dict city. This will erase the key value per city, New York, from the dictionary. Okay, so continue here. What does it matter? Because eventually you want to use in CompuCell things that you define in one steppable, in one class, in another class. Then you may use dictionary. In CompuCell here, each cell object has its own dictionary. Each cell has a dictionary. It was called cell dict, like this. You can use this dictionary to store something new that you want to move around the simulation. For example, for cell in cell list, if age not in cell dict, you create a key age, put it to 0. Okay. First, it is checking if this particular cell here has age as a key. If the cell doesn't have age as a key, then create it and put a 0 value in it. Then change the created value from 0 to add 1 to it, increase by 1. See, if age is in the dictionary of the cell, this second line is not implemented. It goes directly from here to here, and implement 1 in the value. It is already there. So properties attached to cells are automatically shared between the simulations. Okay? This is a very important thing to have in mind, to use a dictionary. There is also another way to automatically share, but this it's more complicated. You have to define a global dictionary like that. You can look at it in the documentation of the CompuCell, in the help manual, CompuCell manual. You're gonna find this there, how to define the global dictionaries. Okay, but for now, let's move on from here. Okay, continues with the MitosisSteppable. This is what I was had to note. Okay, self-parent is the cell which is wandering. It's a cell object that exists at the beginning of your simulation. Okay? We have a single cell, a single object that represents the cell. Then the cell divides. When it divides here, we have the parent, see, I am keeping the semicolon for the cell that I say that continues to be parent, and the child cell is the blue one. And this is a new object, as I said before. The cell is there, but a new object must be created. I mean, the MitosisSteppable created the object, but you have to say what kind of object it is, what are his attributes. This is what with the MitosisSteppable does. Okay, so the parent-child is just a convention to facilitate the coding, actually. Okay, now let's code an exercise in Mitosis. Okay, this is the roadmap. Let's start the simulation from scratch again, because it's easier than modify the previous one, because it involves the new steppable. You have to load from the beginning. So this is the roadmap. Let's go step by step. Okay. The first part, I will not repeat the first slide because we already know. Change the name now to cell division, for example. Grid size 100 by 100 by 1. Use the BlobInitializer, and now you create two cell types. ST, representing the stem cell, and TD cell for terminally differentiated cell. ST and TD. They are here in the left panel, two cell types, ST and TD. And in the cell properties and behaviors, now, besides the growth steppable, you must load the mitosis steppable together. Remember in the contact plugin page, set the neighbor order to 4. I forgot to put this here. This is a new feature of CompuCell, or this version of CompuCell. I forgot to add a figure here about that, but I'm telling you. So finish this, ignore the chemical fields page, and save it. Open again the XML file to modify the BlobInitializer tag. And let's create at the beginning only one single stem cells like this. What should be neighbor order this time? Is 4. Put it to 4. Always to 4. In the simulation, usually to 4. I should tell Maciek to put 4 in the default. Okay, then only one stem cells here. And in the steppable, you adjust, well, the volume target is still 25, but the target volume put, the lambda volume put equals to 5. And add that line to increase the target volume to over 500 now. To the growth be slower. Remember, before it's 25 by 100. Now we are 5 times or slower. To default here, the things are already, I think, are already ready to go. Cell volume, the trigger for the division is 50. The the division is in the orientation axis. Put back the target volume to the initial volume, the initial target. Clone the parent to child. And look here, each cell division results one stem cell and one TD. Always that a cell divides, it creates two different cells, one of each type. Okay? Now save and run the simulation. You should have this. You start with the yellow cell. The yellow is, for me, is the stem cells, and the blue is the TD cells. They start to grow, divide, and goes this way. Now let's change the division trigger. Go to do your steppable and lower the cell volume condition to 30 and run again the simulation. Okay, now the difference now is that the cells grow faster and are smaller, just like that, because when they start with 25, when they reach 30, they divide, and the target goes to 15. They start smaller to grow because you have that division by 2, remember? Okay. let's change a little bit. Let's see what happens if only the ST cells can divide. Well, since we have only one cell at the beginning, we should have only one cell at the end. Okay? To implement the modification to run over the ST cells, you have to replace line 36 for 37 in the screen. Probably your lines are different than mine. So you have to iterate over cell type, but all the cells. Okay, make this modification, you save it and run, and you should have something like that. So you start with one yellow cell and end with only one cell, and all the others are growing indefinitely because now they are not dividing anymore. Only the stem cells is dividing. It is growing, it is dividing, but when it is divided, it always creates a blue cell. So the number of blue cells increase, but the yellow one doesn't increase. Now, see, if you let your simulation run indefinitely, the cells will occupy all the lattice, all the grid, and we will stop to grow. But the target volume of the cells continues increasing because that line in the steppable that is increasing each Monte Carlo step by certain amount continues there. So this creates what we call a pressure in the cells. Okay, this pressure is given by the difference between the actual volume of the cell and the target volume of the cell, and is obtained from the energy term, the volume energy term, like that. Now I'm going to talk a little bit of physics. Okay, so in physics, the intensity, the strength of a force F acting on a system is given by this expression. If you are is a derivative of the potential energy with the distance, with some unit of length. Okay? So for us here, the energy we are messing around when we change the target volume is the volume energy. So there is a variation in this quantity. So this produces this force. Okay. However, pressure is the ratio between applied force on a surface divided by the area where the force is applied. Okay, this is the definition of pressure, force over area. Okay, when you press your finger over your table, you are making a pressure of the table. So when you press, you are exerting a force over an area which is the size of the tip of your finger. So this defines a pressure. Since we have this definition here for force, we can replace force here for this definition. We have the second expression here. But if you look better in the denominator of this expression here, we have area times length. This is volume. So you can write that pressure is the derivative of the volume energy in respect to volume of the function. Maybe complicated, but believe me. So, yeah, but so you can now you can calculate the pressure because we know the expression to volume term, as you saw in the first lecture today. Here, remember, V minus V target is that parabola. So, using this expression here, I can obtain a expression, a real expression for the pressure. So the pressure is 2 times the lambda volume, which comes from here, times V target minus actual volume. So I can measure the pressure in the simulation. The pressure of each cell is subject, is suffering because of the others, because they are compressed in that space. Okay? Let's implement that in the code. Uh, let's use this pressure to control the growth. I mean, when the cells start to fill all the space, we are going to control the growth. We will avoid that the target volume grow even bigger. Okay, but then we have to do the following. Remember, we are controlling the growth according with this function here. V target is increases by a constant amount of each Monte Carlo step. Now we are going to replace this by this G constant quantity by a function of the pressure, which the cell is suffering. And this function here can have multiple functions. Could be linear, could be a fraction like that, can be a real function like that, whatever. You can choose whatever you need from your biological model to implement here and control the growth due to lack of the space in your Petri dish, for example. Okay, let's implement the simple one. The first one, this linear one here. Again, started a new simulation from scratch because it's easier than modify the existing one. Again, the roadmap is there, going. The initial 5 steps are the same as before. We can use 100, 100 as 2 cell types, SCT, TD, BlobInitializer, same plugins as before, contact, volume, center of mass, Steppables, growth, and mitosis, modify the BlobInitializer to use radius 5 and width 5. I mean, you're going to start again with only one cell, one stem cell. And in the steppable file, you put target volume equals to 25 and lambda volume equals to 3 for all cell types. And the growth law at the beginning is 25 over 500. That is what we had in the previous simulation. Now we are going to watch the growth. we are going to introduce, this is the most elaborated model right so far, okay? Let's go by parts, like Jack. Let's this is the initial growth steppable, where we are going to introduce now a plot window like we did before. Remember, from the drop-down menu, in the start, we have to type the start function line because it doesn't have. From the drop-down menu, you go to the scientific plots and set up a plot window, number one, in scientific plots. Configure it here like this or whatever titles you want. And you have to create they give a name to the placeholder of the plot here, AVVol, for example. And what you're gonna look is to keep track of the average target volume of the cells. Okay, see, we are going to start with one single cell, but this cells is going to divide. The division will create many cells, and each cells have growth differently with time because it's a stochastic process. So we are interested in the average target volume of the block for each average target for the cells. And we want to see that value in a graph, how the target volume, the average target value evolves in time. The problem is that we don't have this quantity, average value. We have to calculate it. We have the list of the cells. We have the cell volume of each cell, but we don't have the average value of this. We have to calculate. So in this box here, I'm calculating this. Look, it's quite simple. In line 34 here, I define the average value and set it to 0. And in the variable L, I put the length of the cell list. The length of cell list gives you the number of the cells that your blob has. Okay? So, having this value, iterate for the cell list, for the same cell list here, and increment the AVOL that initially is 0 by the target volume of the cell divided by L. When the sum is over, I have the average value for the target volume of the cell. Okay, then I have the average value here. Line 4, I forgot to tell you, go to the CC3D table, Python table, and add scientific plot, add the data point here from scientific plots, give the name to the same value you define here, AVVol, and plot MCS and AVOL, which is the average vol we have just calculated here. And the code continues as before here. Okay? So This initial code now we can run. Note that see the first box and the third box I type it. The second box and the fourth box were included using the CC3D Python drop-down menu from to edit. Okay. Now you can run the code, save it and run, and tell me what you have. You should have two windows. Yes, exactly like that. Perfect. See, we start with single cell. The cell start always divided in two different cells. The average volume grows when it attains a value of 50. They are cut in the middle, then it start to grow again. This is why we have this zigzag here at the beginning, and continues zigzagging while the cells don't fill the space. But when the space is filled, there is no zigzag anymore. The target volume continues to grow forever here. This is what causes the pressure. Okay? And since our growing is linear, so here we have a straight line here. So let's fix this. Yeah, the system gets froze because the cells cannot move anymore. So it just oscillates around this center of mass position, but doesn't go anywhere. Don't divide nothing. Okay. Now we are going to keep track of each cell pressure by adding a dictionary entry, because pressure is not by default attribute for a cell from the beginning, like volume, surface, ID, type, pressure, it's not. This is why you have to use the dictionary to attribute a new thing to the cell. So we are going to attribute this dictionary. This is in the constant initializer Steppable. There, where you have in the very beginning, when you have the cell target volume, cell lambda volume, you type this line, cell.dict, how do you say this? I don't know this. Brackets, pressure between commas equals to 0. At the beginning, everything starts with no pressure. Okay? And then, sometimes my mouse goes away. We're going to add a second plot window. See, it's a really new plot window. It's not a second placeholder as we did before. Again, go to start function, go to Python drop down menu to edit scientific plots, set up plot window. Again, configure it with a different name, a different color. For example, here the first one are dots, this one are lines, just to be different. And always to this end, let's use a bigger size. Puts always equals to 5. Okay, once the window is created, the plot window is created, we now have to calculate the same way we did with the volume, the average pressure. Okay, so this is done in this line. We have we define average pressure variable here and put equals to 0. And inside the same interaction for the cells, we put the cell dictionary for that cell equals to the uh, expression we just I just showed in the physicals class few slides ago. 2 times lambda volume, cell lambda volume times cell target volume minus cell volume divided by L, and divided by L to increment the average value. After the loop ends, I will have, besides the average target volume, I will have the average pressure of the cells in the system. Don't forget to include line 44, which is the line which really adds the plot to the graph window. If you run the simulation, that's what you have. Both targets volume and average pressure are growing indefinitely because there is no cutting, no threshold for stop growing. How do you fix that? Let's fix that. Let's implement the function to make a threshold in the growth. Let's choose the first one, as I said, the linear one. So we have here the behavior of this function. This is a negative linear. And we introduce this by changing the growth rate by this expression here. Look, this FP always gives us a value that can be positive or negative according with this function. Okay? But I'm going to use only the positive values here. This is what I'm doing here. I'm multiplying the growth that I had before, 25 over 100, times the max. Max is a function which gives you which value is bigger among these two values inside the parentheses. Okay, so if p ref minus cell dictor is negative, gives you 0. If this quantity is positive, gives you this quantity. Okay? Understood? So this is a threshold. If the pressure becomes too high, I mean, see, if the cell pressure is higher than the reference pressure, this quantity becomes negative. Then there is no growth. The max function gives you 0, and the target doesn't grow. This is what this function does. Let's see. If you include this line, put it to run, you should have this result. Look, both average target and average pressure tends to stabilize it with this control here. And the amount of of stabilization, how big should be the average pressure. It depends of the reference value you put in the function, for example. You can change this to see how these values can change. The next slide, it's a homework, let's say. You can try the different functions here to see what kind of behavior you have in the average target volumes and pressures. And even you can change in the Hill function here, you can change the n exponent, you can change the k, whatever. You can play a little bit to see how to manipulate that. So I think that's it for now, and thanks for your attention.
So I'm not sure which paper we are talking about. I mean, it's relatively easy to write your own finite state model. If you have this finite state model implemented, for example, in MabBoss, that works. So I'm not quite sure which example of finite state model you're referring to, but there's nothing preventing you, if you have a package that implements state transition models, from using those. Actually, we just are in the process of publishing something called CompuCell3D, that is a Python extension library to implement finite state models. It's been a while. There was a bit of a delay in getting that submitted for publication. It is available on archive, and so you could look at it there, and the code is available. So the person who's leading that project is Michael Getz. Paul Macklin also wrote a simple Hill function rate equation rule grammar. And these days, with coding assistance, one could probably re-implement that in a Python library without too much effort. That would be an interesting thing to add to CompuCell as well. Remember again that anything that is a Python library can be included in CompuCell relatively easily. So if there is a Python library that does what you want, that's fine. The one thing is, if you're using a Conda environment, you're going to have to then rebuild the Conda environment with that library included. That CompuCell3D library is available for download if you want to try it out. There's a GitHub link in the paper that'll take you to the download for it. It was originally written by Giuliano Gianlupi, and he is now working as a developer in a biotech company, and so he doesn't have too much time to work on CompuCell anymore. Well, thank you very much, everybody, for coming. We'll see you tomorrow.